I think we are, yes, slowly everybody's connecting. So welcome back to this appointment, as you know, every first Tuesday of the month. And just let me share the screen to remind you that this is an initiative co-led by the Carlo team, so led by Amanda Sierra, and with the help of Marta Pereira and Federico Soria, and the UNIL team with myself, Claire Compagnon, Fanny Martineau, and Giorgio Corsi. We have a very rich calendar, as you see already here, until the end of this year, but just as a reminder, in case you miss some of the live appointment, you can still follow and attend the seminars offline. This is the link. So all the seminars are recorded. You can find them here. And today we have the pleasure to have Alain Bessis with us, who will be introduced by Giorgio Corsi, and the Q&A will be led by Federico Soria. So without further ado, Giorgio, I leave to you.
Thank you, Rosa. Hello, everyone. It's a great pleasure for me to introduce today Dr. Alain Bessis. During his PhD, Dr. Bessis studied the transcriptomic regulation of the nicotinic acetylcholine receptors. Then he moved to the function of the same receptor in higher brain functions. Since 2002, he studies how microglia interact with neurons under physiological conditions with a strong focus on synaptic function. Currently, he is a research director at the National Center for Scientific Research in Paris and professor affiliated to the École Normale Supérieure. Please, the floor is all yours. You can start sharing.
Yeah. So, can you see the screen? Yes.
Yes.
Okay, so hello everyone. Thank you, Giorgio, for the introduction. Thank you, Rosa and Amanda, for this invitation. I'm super honored to be here after so many great speakers. And I'm super glad to talk in front of such an audience. And I guess in front of this audience, I don't have to make a big introduction on microglia, because we are all convinced that these are super interesting cells. I would like just to start by showing this pie chart that I like to show to my friends working on astrocytes. That shows that in the cortex, microglia is super dense. And here, I guess everyone knows this nice figure of Rosa's paper showing that microglia have multiple functions in physiological brain. And in the lab, we are working on I should say maybe here a super provocative idea that in the brain also neurons are useful. And so we are studying the interaction between microglia and neurons. And more specifically, the most neuron-specific function, I should say, is the synaptic function, I think. So this is why we are studying how microglia and synapses are interacting together, both excitatory and inhibitory synapses. And today, I would like to show you the result we had on how this microglia synapse interaction is regulating during sleep. And there are several reasons to that. So first, on the microglial side, the fact that microglia sense and react to the sleep-wake state of the brain is known since a long time. You see here that it is known that the morphology of the microglia is changed, and we all kind of agree that morphology is a kind of proxy to talk about function of microglia. Recently, the group of Olivier Pasquale also showed that the motility of the microglia is regulated during the sleep-wake cycle, and super recently, like last month or 2 months ago, there was a nice paper showing that actually microglia regulate the sleep-wake cycle through P2Y12 and norepinephrine. And on the top of that, it has long been known also that the microglial signaling molecules were differentially produced during the sleep-wake cycle. And you see here, for instance, that TNF is highly produced during the light period, which is the sleep period of the mouse. On the synapse side, the fact that synapses are regulated during normal sleep-wake cycle is also known since a long time. For instance, it is known that during wake, synapses, excitatory synapses are potentiated, meaning that there is more receptor accumulated at the synapse. And during sleep, they are downscaled, and the Tononi and Cirelli have raised this nice hypothesis that actually during learning, during wake, there are learning processes which induces this potentiation, and this is the role, one of the role of sleep, to induce the downscaling of synapses to brain oscillation, allowing a new learning to occur in the next sleep-wake cycle. And there is more about synapse and sleep. For instance, it has been shown that during the sleep-wake cycle, there is a huge change in the phosphorylation of synaptic protein. You see here that like about 800 proteins see their phosphorylation pattern cycling during the sleep-wake cycle. And there is more because it has been proposed that the phosphorylation status of 80 synaptic protein that they called SNPs, synaptic sleep need index phosphoprotein. So the phosphorylation status of these 80 protein define encodes the sleep need. So there is a strong relationship between between synapse and sleep. There have been interactions shown between microglia and synapse during sleep. And for instance, in the same paper, the group of Olivier Pasquale showed that the distance between microglia and spines, so microglia and excitatory synapses, changed during the sleep-wake cycle. And also the group of Cristina Limatola and Agnès Najjar showed that this change in the potentiation of synapses during the sleep-wake cycle is actually dependent on microglia, because you see here that in the brain of microglia depleted mice, there is no more cycling of the potentiation or on the synaptic strength of excitatory synapses. And this is most probably due to a CX3CR1 signaling, because this cycling does not occur in this mutant mice. So the relationship between microglia and synapse has been studied already, but mostly, as you saw, on excitatory synapses, and the mechanisms were not super well characterized. So we decided to investigate how microglia could regulate the inhibitory synapses during the sleep-wake cycle. And we focus on the layer 1 and layer 5, because in layer 1, most of the synapses are due to somatostatin interneurons, whereas in layer 5, most of the synapses, the inhibitory synapses, are due to PV interneurons. And we used immunohistochemistry to quantify the amount of receptors, of GABA receptor, that are accumulated at synaptic side, defined by the apposition of gefferin, the scaffolding protein, and VGAT, the vesicular transporter of GABA. And what you can show here is that in layer 1, we did see an increased accumulation of GABA receptor in the layer 1, but nothing at the extrasynaptic site. And we didn't see any change during the sleep-wake cycle in layer 5, so most probably the PV interneurons. Of course, the first thing we did was to show that this regulation was due to microglia, and it did, because you see that there is no more regulation, no more increase in the GABA receptor in microglia depleted mice. So I showed you that during the sleep-wake cycle, microglia seems to regulate the inhibitory synapses specifically located in layer 1. And of course, this can be due to the sleep-wake cycle or the light-dark cycle. And we wanted to show that we have here a sleep-dependent mechanism. So we induced sleep deprivation to mouse and measured the accumulation of GABA receptor. And as you can see here, upon sleep deprivation, there is no more increased GABA receptor at synapses in layer 1, showing that, yes, this is a sleep-dependent mechanism that is here that allows microglia to regulate the GABA receptor in layer 1. Of course, we wanted to go a bit further in the mechanism of this regulation, and you know that sleep comes in two flavors. There is the non-REM sleep and the REM sleeps. The REM sleep that can be distinguished by EEG. And the group of Wen Biogan showed that in the layer 1, there are these NMDA-dependent spikes that occur and that are involved in the plasticity of spines during sleep. So we thought that maybe they could also be involved in the regulation of inhibitory synapses, the strength of these synapses in the layer 1. And so we tested to deplete the REM sleep in mice. To do so, we recorded the EEG in live animal, and as soon as they enter in REM, we just woke them up. And you see that this is very efficient because this induces an 80% decrease in the amount of REM sleep. And as you can see here, in REM sleep deprived animals, there is much less GABA receptor at synapses, showing that, yes, there is a REM sleep dependent mechanism that triggers this increase in the GABA receptor in layer 1 dependent on microglia. We, of course, we think that these are NMDA dependent mechanism, but at this stage, we are not sure. We wanted then to identify the molecular mechanisms linking microglia and inhibitory synapses in layer 1. And to do so, we switched to an ex vivo model to brain slices using this paradigm of so-called inhibitory LTP that was found by the group of Michael Higley, who showed that a short stimulation of NMDA on acute slices induced actually an increased synaptic strength, specifically in somatostatin interneurons, which synapses are located in layer 1, but did nothing in somatostatin synapses, so mimicking what we have found during the sleep-wake cycle. So we thought that maybe this could be the same kind of mechanism. And in addition, this group showed that the link between excitatory synapses and inhibitory synapses is CAM kinase, which are phosphorylated upon stimulation by NMDA. So there is a stimulation of NMDA that induced the phosphorylation of CAM kinase, which induced the increased accumulation of GABA receptor. And we tested if the mechanism could be the same during the sleep-wake cycle, but first we identified the involvement of microglia here in this mechanism. And so we did the same thing. We used organotypic slices and analyzed the synaptic content of GABA receptor using immunohistochemistry. And as you can see here, there is the same increase at synapses, nothing at extrasynaptic site, and nothing at layer 5 inhibitory synapses. And of course, we showed that this was dependent on microglia because there is no effect when microglia are depleted. So then we had the system to study the mechanism, the molecular mechanisms of this regulation. And we tried the first thing, as everyone, the CX3-CL1 pathway, but as you can see, using a CX3-CL1 blocking antibody, we could not prevent this the effect of ILTP, the increase of GABA receptor or synaptic GABA receptor, but using neutralizing TNF antibody, we could prevent this ILTP, this accumulation of GABA receptor, showing that yes, TNF is the signaling molecule involved in this regulation. Of course, there was a kind of debate, I think, on the site of expression of TNF, but in all the published single cell transcriptomic studies in the brain, you see that TNF is only found in microglia, expressed by microglia. And we wanted to, but there are other papers showing an expression or at least immunohistochemistry of TNF outside microglia, and we wanted to investigate on that. And I show you this image as a, I don't know, a cautionary note maybe. Using antibodies that were used in these studies, we could show that we could indeed see immunoreactivity outside microglia. But as you can see here, when we tested the same antibodies in full TNF knockout, we had exactly the same immunoreactivity. And I must say that we tried a lot to see, to understand this non-microglial expression of TNF that is often published, and we consistently found that the antibody used in this study gave the same signal in full TNF knockout as in wild type, so suggesting that this non-microglial expression of TNF might be a background. Okay, so we think that microglial TNF is involved in the regulation of GABA receptor in layer 1. We confirmed this using microglial specific knockout of TNF, and you see in this mice there is no effect of application of NMDA, and we further show that using neutralizing TNF receptor antibody, that there is no effect when we block the TNF-R1, but blocking TNF-R2 has no effect. So this shows that TNF is required to regulate the GABA receptor at synapses, but it is also sufficient because we apply TNF on these slices. You see that it increased the synaptic GABA receptor, but not the extrasynaptic GABA receptor. So you remember that the group of Michael Eagley showed the involvement of phosphorylation of CAM kinase, and actually when we induce this LTP, we also showed the increase of phosphorylated form of CAM kinase using immunohistochemistry, but we could not see it when we depleted microglia or when we blocked the microglial TNF, using a neutralizing antibody. So this shows that most probably microglial TNF is involved in the regulation of the phosphorylation of CAM kinase, allowing the synaptic accumulation of GABA receptor. So of course, the next step was to try to understand the backward signaling, how neurons signal to microglia to tell them to do something at inhibitory synapses. And of course, the first candidate is ATP, because there have been a lot of studies showing that ATP signal to microglia to do something. And we actually demonstrated that, yes, ATP is involved in this regulation, because when we degrade ATP using epirase, as you can see here, we prevented this increased synaptic GABA receptor, and we tested the P2 receptors. We all know that there are a lot of receptors expressed by microglia, and the first candidate, of course, was P2Y12. And as you can see here, using an antagonist that is not written, but it's an antagonist of P2Y12 receptor, there were no effect. So it's probably not P2Y12 receptor that is involved here. But when we block the P2X7 receptor using this antagonist, you see that it prevented the synaptic accumulation of GABA receptors. So most probably P2X7 is involved. And again, it is required, but it is also sufficient, because when we stimulated the slices using a busy ATP, which is an agonist of P2X7 receptor, you see that it mimicked the effect of LTP, or it mimicked the effect of application of NMDA. And also, we could also show that this busy ATP also induced the phosphorylation of CAM kinase, suggesting that, yes, this is the same pathway. And talking about discussion on expression pattern, there is also a kind of debate on the expression pattern of P2X7, but this time I think it's kind of legitimate, because it seems that, yes, P2X7 is expressed by microglia, but it is also expressed by several other cell types of the brain. So we wanted to be a bit more sure that we were talking about microglial P2X7, and we showed that if we apply BZ-ATP but without microglia, there were no effect. So P2X7 is actually acting upstream from microglia. And similarly, BZ-ATP has no effect when TNF is depleted from microglia, or when CAM kinase is inhibited. So we think that P2X7 acts on microglia, or maybe in a cell that is upstream from microglia, to regulate the synaptic accumulation of GABA receptors. Okay, so what I've shown to you up to now is that in this ex vivo model, the stimulation of the slices by NMDA induced the recruitment of microglia through the P2X7 pathway, leading to the production of TNF, which regulates the inhibitory synapses in layer 1 through a CAM kinase dependent mechanism. So this was in ex vivo, but you remember that the idea was to do that, to understand the mechanism occurring during the sleep-wake cycle. And oh yes, sorry. And I just want to say that we have not demonstrated that astrocytes are not involved. So they may be involved either upstream of from ATP or downstream of TNF. But at this stage, we don't know if they are involved. So we wanted, as I said, we wanted this. This experiment in slices were to try to understand the mechanism occurring during the sleep-wake cycle, and we went back to the sleep-wake cycle and demonstrated. So you remember that during sleep there is more accumulation of GABA receptor at synaptic sites. This accumulation is prevented by PLX, so by depleting microglia, but it is also prevented when microglial TNF is depleted, specifically in microglia and in P2X7 knockout. And there is this involvement of CAM kinase, and we could show that during the sleep-wake cycle, there is this increased phosphorylation of CAM kinase, which is prevented in microglial TNF knockout and in P2X7 knockout. So we think that during the sleep-wake cycle, the mechanism that is involved here is the one that we have also described in this organotypic slice. Okay. So what I've shown to you up to now is that microglia are able to regulate the excitatory and inhibitory synapses. So the excitatory synapses apparently through the CX3CR1 pathway, and the inhibitory synapses through the TNF pathway. We next wanted to study the consequences of this regulation. So how microglial TNF, in our case, control the sleep-wake cycle, or the functions associated to sleep. So we first studied how it it regulates the sleep-wake cycle itself. By the way, the same paper showed that in CX3CR1 knockout, there is a change. There is more non-REM sleep, suggesting that microglia regulate the sleep-wake cycle through the fact that they regulate excitatory synapses. But we did the same thing in microglial TNF knockout. We analyzed the structure of sleep. And as you can see here, there is no difference in the amount of wake or non-REM or REM sleep, except maybe a small difference here, suggesting that this pathway, the fact that microglia regulate this inhibitory synapses, is not really involved in the regulation of sleep-wake cycle itself. And by the way, this was also shown a long time ago on full TNF knockout. This recapitulates exactly what was found earlier. So this shows that most probably microglial TNF is not involved in the regulation of sleep-wake cycle. So, but what could be the function of this regulation? And we analyzed the brain activity during the sleep-wake cycle in control and microglial TNF knockout. And as you can see, the activity is not different in wake or in REM sleep. And in non-REM sleep, there is a small difference here in the slow wave part of the spectrum, the delta band. And this was kind of interesting to us because it is known that these slow waves are an alternation of so-called off period and on period during which there is a synchronization of a silent and active phase of the neuron of the cortex. And if it is interesting for us, it's because the group of Tononi has already shown that these downstates are induced by the activity of the somatostatin interneurons, which are most probably the neurons that are involved, that are regulated by the microglial TNF. So the hypothesis here was that if microglial TNF controls the strength of the inhibitory synapses made by somatostatin interneurons, most probably it will regulate the slow wave themselves. And we have tested this hypothesis by extracting the slow waves from frontal EEG in control and microglial TNF mouse. And as you can see here, we found that the slow waves are changed in this microglial TNF knockout with a longer duration and lower slope. And the slope is important because this is the moment, the exact moment when neuron change go from active to down states. Okay, so microglial TNF regulate the shape of the slow waves, and we think that this could be important because it is known also that the shape of the slow wave actually is important for memory consolidation. This is shown in this experiment where a subject have to learn a sequence of finger tapping, and they learn in a certain speed. And after a night of sleep, you see that they perform a bit better. And this is the so-called memory consolidation. Wow. This is a so-called memory consolidation. And if during the sleep, the subject hear a sound as soon as they enter into sleep, as soon as there is a slow wave in their EEG, you see that this induces a change in the duration and the slope of the slow wave. And the consequences in green here is that there is no more memory consolidation. So in this motor task, the shape of the slow wave is important for memory consolidation. And this can be tested also in mice. If the mouse learn to run on a complex wheel as you can see here. You see that they learn after different trial session, and after a sleep period, you see that there is a consolidation, meaning that they start better. They are better than the level at which they finished the last session, and this is what is called consolidation. And this consolidation depends on sleep, because if you sleep deprive the mice, you see that there is no more consolidation on this task. So we did this experiment on control and microglial TNF knockout mice. And what you can see here is that this TNF knockout mice learn well to run on this rotarod. And the first day and the second day, there is no difference between the control and the TNF knockout. But as you can see here, the mouse with no TNF in microglia do not consolidate the learning process that they have done during the first day. So it seems that microglial TNF is involved in the consolidation of memory in this motor learning. I won't go into much detail, but we have done other memory consolidation tasks. For instance, this task in which the mouse explore two objects the first day and the same object the second day, but one of them is on a floor with a different texture. And this, of course, the second day, the mouse explore more the new floor, but when there is sleep deprivation, they do not distinguish the two objects. And we have done the same thing in the microglial TNF knockout. And what you can see is that there is also an alteration of the memory consolidation in this mouse. Just to say that not all the type of memory consolidation are affected by the depletion of TNF in microglia, because you can see that in this task where the mouse has to remember the place of objects. So there are two objects, and the next day you put a new object here, and the mouse explore more the novel object on the second day, except when they do when there is sleep deprivation. And in the case of microglial TNF knockout mice, they do not discriminate the new object. It's not different. Sorry, it's not different between control and microglial TNF. So not all type of memory consolidation are dependent on microglial TNF. And of course, we have done a lot of other behavioral tests that are not different between control and microglial TNF knockout. Okay, so what I've told to you up to now is that microglial TNF are involved in the regulation of inhibitory synapses in the upper layers of the cortex by a CAM kinase dependent mechanism. Microglial TNF also control the slow wave in the cortex and is involved in the memory consolidation of some, but not all, learning tasks. Of course, we do think that there is a causal link between these three aspects, but we, as you can notice, we have not demonstrated the causal link yet. During the last minute of this talk, I would like to finish by another study that we have done linking microglial TNF and sleep, wake, and maybe synapses, which is based on the observation that you see that CAM kinase, the phosphorylation of CAM kinase, seems to be regulated by microglial TNF. And this is important because you remember that the phosphorylation status of protein was massively regulated, for synaptic protein was massively regulated during the sleep-wake cycle. And because we have seen that microglial TNF regulate a major kinase, the CAM kinase, which is a synaptic kinase, we thought that maybe TNF could be involved in this regulation. And also, you remember this AT protein that were supposedly regulated the sleep need. Well, one of these proteins is also the CAM kinase. So we thought that maybe we could study how microglial TNF could regulate and could be involved in this cycling of phosphorylation in protein. So we studied the proteome and phosphoproteome of control and knockout mice in sleep and wake, and compared the result. Here, this is the comparison of the amount of protein in that we could find in the cortex of control and knockout mouse. And as you can see, there is not a lot of difference. Only 7% of the protein are their level differentially regulated between control and TNF mouse. And during sleep, this is about the same thing. Only 6% of the protein have different level between control and TNF knockout, and the amplitude of the change is very low. So there is not a lot of thing happening at the level of the expression of the protein. But when we looked at the phosphorylation of this protein, then the things were a bit different. You see that concerning the phosphorylation, only 7% of the protein have their phosphorylation dependent on microglial TNF during wake. But when we compared what happened during sleep, you see that about 1,500 protein have their phosphorylation that depends on microglial TNF. And we tested if this change was due to, and half of them are dependent on TNF. And as you can see here, the amplitude of changes are huge. So this shows that during sleep, TNF massively dampens the phosphorylation of the protein under normal sleep. And we investigated if this change were due to sleep or to the light dark cycle. So we induced sleep deprivation and measured also the phosphorylation of this protein. And as you can see here, about 60% of this protein were indeed sleep dependent. The phosphorylation was sleep dependent. So they were both sleep dependent and TNF dependent. We then analyzed the so-called pathway of this TNF dependent molecule, and strikingly, almost all the pathway were linked to the synaptic function. So this was a bit puzzling, and we went back to this cycling protein, this 800 cycling protein, and analyzed how they depend or not on TNF. And as you can see here, almost 500 of them has their phosphorylation that depends on microglial TNF. And what about this 80 synaptic protein that controls the sleep need, that are supposed to control the sleep need? So 80 protein, and almost all of them have their phosphorylation that depends on TNF. So if this is true, if this 80 protein really control the sleep need, we expect that in microglial TNF knockout, the sleep need should be altered. So we have tested this hypothesis. And to do this, we induced sleep deprivation to control and knockout mice and analyzed the homeostatic sleep. First, we measured the activity in the cortex, and it is known that the slow waves during non-REM sleep is a kind of measure of the sleep need. So when you sleep deprive a mouse and you let it sleep, you see that there is a huge amount of slow waves that slowly decrease when the mouse sleeps normally. And of course, after sleep deprivation, there is a sleep rebound, so that the mice are less in the wake state, and they are more in sleep. And when we did the same thing in microglial TNF knockout, as you can see here, there was less slow wave, meaning that the sleep need is decreased in this microglial TNF knockout in agreement with the regulation of this 80 SNP protein. And strikingly, there is no sleep rebound, as you can see here. The mice sleep the same amount as the control, and this is shown also here. Okay, so this is it. What I've shown to you I'll try to synthesize everything I've told to you today. During the basal sleep, microglia is able to sense the sleep through maybe REM sleep and ATP, and in response to the sleep, microglia produce TNF that regulate the phosphorylation of hundreds of synaptic proteins. These synaptic proteins do not change the basal sleep, but instead they kind of regulate what seems to be sleep functions, such as synaptic homeostasis of memory or memory consolidation. But when there is a sleep need during enforced wake, then the part of this phosphorylated synaptic protein are involved then in the regulation of what is called homeostatic sleep. Okay, I'm done. I would like now to thank Maria Pinto, who is the postdoc who did most, if not all, the work that I have presented. She's a super brilliant postdoc who is now back in Portugal. And fortunately, there are now new super postdocs in the lab. The sleep analysis and the recording have been done by Clément Léna, the group of Clément Léna and the group of Véronique Fabre, which I would like to thank warmly. And so I'm done, and I'm of course open to discussion.
Okay, Alain, thank you for this great talk. We open now for the question and answers. I think the first one is from Amanda, actually. Amanda, go ahead, please.
No, but I can wait until the end.
Okay, so the next one is from, I have one written here from Petros Ligoshigakis. I hope I pronounced that correctly. The question is, he said, hi, great talk, thank you. What happens or what you predict would happen in chronic TNF-alpha activity? Well,
yeah, of course, because in a lot of pathology, there is a huge production of TNF. So we expect that all this pathway is altered in this kind of pathology. But I must say that this is a very difficult question, because in the control of synapses, it has been shown that depending on the concentration of TNF, the results can be completely different, can be sometimes opposed. So at low concentration, TNF can do something, but at high concentration, it can do the opposite. And so I don't know what to answer. I don't have any prediction. And in addition, we don't really know how TNF is produced in the brain. We know that microglia produce them, but we don't know how, and we don't know if there is different status of TNF production under basal activity or under chronic pathological situation. So I'm sorry, but I don't know. I cannot predict anything.
Okay, the next one is from James Woodward. You raise your hand. You can open your mic.
Really interesting talk, thank you. I was just wondering, do you think this could potentially complement the TLR9 finding that recently came out, which showed that activation of that inflammatory pathway, and which then triggers NF-kappa B and TNF for long-term memory formation? Do you think this sort of complements that work, or do you sort of see this as being quite independent? And yeah, what are your views on that?
Yeah, thank you for the question. I must say that I don't know the paper you are talking about, but you said that it is under pathological condition, and yes, I do think that what we are looking at is not pathological condition, is really basal sleep, physiological conditions. And yes, I do think that these are completely different mechanisms, because again, the concentration of TNF is super low at basal condition and extremely high in pathological conditions. And I'm sorry, I don't have any other comment or any further data to support that. But yes, I do think that these are complementary situations. Sorry not to be able to be more precise in this answer.
No worries. Thank you.
Okay, I think Amanda, you're next because there are no more questions in the chat field.
So first, I want to say thank you, Alain, because you have an amazing story there. I really love that you were able to show that this link between slow waves and memory consolidation and the microglial TNF-alpha. So I thought that was very in spite of not having a global effect on the sleep cycle. So I wanted to ask you, do you think there are, you've shown TNF-alpha and you've shown microglia, but the effects on memory consolidation and slow waves are not necessarily linked to the glutamate or receptors or GABA receptors that you were analyzing, right? So the thing is that your story made me think of the lymphatic system, that it's also regulated during sleep, and it also has a role in memory consolidation. So you think it's possible that some of these microglial TNF-alpha is not directly acting on synaptic connectivity, but more on like drainage of waste. What do you think about that?
Yeah, thank you, Amanda. I must say that I fully agree with you. We have not shown a causal link. This is why I told that at the end. We are conscious of that. There is no causal link. It's just we think the most probable link that because as I said, the memory consolidation depends on slow wave. The slow wave depends on inhibition in the upper layers. So we think that these are linked. But yes, sleep is such an integrated mechanism. I'm convinced that if you if you alter the lymphatic system, you just mess up everything. So I cannot say more than I agree with you that yes, most probably there are other ways, other pathways that are impacted when you deplete microglial TNF.
Okay, thank you.
Okay, next one is written in the chat field from Colm Cunningham. It says, very interesting talk, and I know you have shown nice in vivo data too, but have you looked at how TNF is produced by preparing slices per se? That is, slicing brain tissue is a substantial acute brain injury, so basal TNF is not really basal. Just wondering what your thoughts are about this.
Yeah, this is a super relevant question, of course, and we are all extremely concerned when we are using these organotypic slices. We are waiting at least 10 days after the slicing procedure to measure to do the experiment, and when we try to quantify the TNF in these slices using ELISA, it is extremely difficult. So I guess that there is not a lot of production. The production is not super high. So we do think that after 10 days, we are at basal level. This is what I can say. For instance, to validate the fact that the TNF the knockout is done, we could not measure the TNF, the level of TNF in slices. So the level is too low, even using a super efficient ELISA test. So we had to go to cell culture. So this is why I think that the level of TNF is extremely low, even in these slices.
Okay, now is Trevor Owens. You raise your hand.
Yeah, very interesting talk. I hope you can hear me. You just commented on the low level of TNF, and I know you showed some data somewhere in there about TNF receptors. So the question is this just microglia modifying the local environment by secreting TNF, or must there be microglial neuronal interaction? And are these specific subpopulations of microglia that interact selectively with these neurons?
I wish I could answer.
Sorry, yeah.
No, this is the most fundamental question that we tried to answer, but I must say I don't know. What I know is that the group of Microglia, when they studied this inhibitory LTP thing, they showed that if you inject phosphorylated form of CAM kinase in the neuron, you only regulate the inhibitory synapses done by somatostatin interneuron. So it means that you can massively regulate everything, and only the somatostatin synapses will be regulated. So we don't need here a super local microglia neuron interaction with TNF. A global interaction is enough. But unfortunately, we are not able to visualize TNF. We have tried very hard, but we could not visualize TNF in these slices in the brain.
Yeah.
So, yeah.
Okay, thank you.
Thank you.
Okay, now Rosa.
Yeah, thanks, Alain. Beautiful talk, because I'm very much in line with Trevor's question. I was thinking about what is your take about this heterogeneity, if there are microglia, I don't know, even by probably, I don't know if you've tried by in situ to see if there are certain cluster of microglia producing TNF-alpha over others. And what is your explanation about this layer specificity? Or...
Yeah, no, but this is such an important question. Of course, we would love to answer to it, but again, we have not been able to see a specificity, to understand the origin of specificity. I think this is a neuron endogenous property, because, as I said, if you inject CAM kinase, phosphorylated form of CAM kinase in all the neurons, only the synapses located in layer 1 answers, so it's neuron autonomous, but we have not been able. I don't know if anyone here in this audience know how to visualize this TNF in the brain. I would love to have some input, but really we could not. I don't know. I don't even know what is the mechanism of the TNF production. I have no idea.
That will be the next challenge to address.
Okay, we don't have any more questions. I don't know, Rosa, if you want to close the session, or...
We can close it together. And thanks again, Alain. Beautiful talk. And thanks to everybody being here. See you.
Okay.
Bye.
She had an unstable connection, so she was saying goodbye, but we lost you for a minute. It's Rosa.
Okay,
so thank you
again. Thank you again. Bye.
And see you next month.
Thank you, Alan. See you next month. Bye.
Bye. Thanks a lot.